EP 120, Lab 3, Translation and DNA Fingerprinting. So, as I mentioned before, the DNA is the important blueprints for all the proteins needed for the body, and it stays safe in the nucleus. And then a copy of specific parts of those blueprints gets sent out as messenger RNA to go to the ribosome, which is the enzyme complex that leads to the production of proteins, all of this occurring in the cytoplasm. So again, DNA replication occurs in the nucleus, nice and safe during the S phase of the interphase. Transcription is making the messenger RNA copy of one gene only, so just a tiny part of the DNA. And what does a gene code for? What does a gene code for? Chromosome, protein, tRNA, amino acid, nucleotide. I hope you chose protein. And then translation. Translation occurs in the cytoplasm. It is when the ribosome uses the information on the messenger RNA to make a specific protein. So, genetic code. Why do we call our DNA the genetic code? Well, it turns out that there are 20 amino acids, and as we know, only 4 different nucleotides in the DNA. So it was crazy for the scientists to try to figure out how you're able to turn this 4-letter sequence into 20 different options. They finally figured out that each amino acid has at least one 3-letter codon assigned to it. So 3-letter code is assigned to each type of amino acid. So for example, in DNA, if your code reads TAC, I mean adenosine, that will get turned into messenger RNA, that's AUG, and AUG is the codon that codes for methionine amino acid. You don't have to remember which amino acid is being coded for, just that it's a 3-nucleotide code in the messenger RNA called the codon that gives you a specific amino acid. And so here is the messenger RNA sequence for some gene. And so underneath it are these individual letters representing amino acids. So you can see how you get amino acid sequence from the codons in the messenger RNA. So sequence of codons in messenger RNA determines the order of amino acids in the protein. Codons in messenger RNA yield amino acids in the protein. So translation. Translation is turning the genetic code in 3-nucleotide chunks or codons into the language of amino acids, translating from nucleic acid to amino acid. This is used to make protein, and you need the ribosome. You also need the messenger RNA, because that is carrying the message, has the coded message. You need tRNAs. tRNAs bring the appropriate amino acid to the ribosome for producing the growing protein chain. And you need the ribosomal RNA because it is part of the ribosome. Ribosomal RNA is a key part of the ribosome. And what do ribosomes do? They make the proteins. Translation. So here is the tRNA. tRNA has a 3-dimensional structure, and at one end is the amino acid, and at the other end is a 3-nucleotide sequence called the anticodon. And what happens is the anticodon complementary base pairs to the codon. That way, the tRNA knows it is carrying the correct amino acid. So each anticodon sequence has its own amino acid associated with it. So for example, if the messenger RNA reads UUA, then the tRNA's anticodon is AAU. U to A, U to A, A to U, UUA, and this would code for the amino acid leucine. Again, you don't have to remember the specific sequences, just that the tRNA anticodons complementary base pair with the messenger RNA codons, and that's how they make sure the correct amino acid is being used. So, messenger RNA is the template for the ribosome. It has the sequence. The ribosome attaches and connects to the messenger RNA. Messenger RNA, ribosome in blue. Oops. DNA is, of course, permanent. That's why it hides in the nucleus. RNA is temporary. So the tRNA comes in and binds to the codon via its anticodon and brings in the correct nucleic amino acid to attach to the growing amino acid chain. So here is the ribosomal RNA in brown. Here is the messenger RNA. Here is the ribosomal RNA is part of the ribosome. The messenger RNA, of course, carrying the sequence, including codons such as AUG. And then the tRNA, or transfer RNA, has the anticodon that complementary base pairs with the codon and is carrying the amino acid. So then the next tRNA comes in, that complementary base pairs to the next codon, and then its amino acid will attach to this one, and the ribosome will keep moving down the messenger RNA, bringing in more and more and more. I really recommend you watch one of the videos that I have on the Canvas page that shows videos of the translation process happening. All right, something we need to be able to do is use this lovely chart to turn nucleic acid sequence into amino acids. So say, for instance, you start with DNA sequence. Well, this chart uses RNA. The reason why we know that is because look at these U's, lots and lots of U's. So that means we need to turn the DNA into RNA. So we do this by complementary base pairing. Complementary base pairing. What base pairs with T? It's going to be A. OC, G. OG, C. A, complementary with T. A is the first letter of the codon. So that means the amino acid is somewhere in this strip of this chart. G, complementary base pairs with C. G is the second nucleotide in the codon. So we use this region here going down, and so where the two overlap, the amino acid is in this area here, and then the third nucleotide, C, again complementary base pairs with G. Over here, C, third letter. Now we know the amino acid is serine. So AGC, that codon translates to serine, the amino acid serine. Now, you do not have to memorize this chart. It will be provided for you, but you will have to be able to use it for quiz and for the Unit 1 exam. So make sure you understand how to use this lovely chart. First letter in the codon, second letter in the codon, third letter in the codon. All right. Turns out that 99.9% of DNA is the same in all humans. Crazy, huh? Even though we seem so diverse, genetically we are pretty identical. However, one way that there is difference in sequences is in locations where restriction enzymes can cut the DNA. So a restriction enzyme is a type of enzyme that looks for a specific sequence in the double-stranded DNA. When it sees that sequence, in this case GAATTC, then it is able to cut the DNA, breaking it up into multiple pieces. So here we have restriction enzyme. It sees its sequence of GAATTC and some double-stranded. It cuts along this line. Boom, now we have two pieces of DNA. The restriction enzyme, found another sequence that it recognizes. It then again cuts the DNA, and look at that. This piece of DNA got cut into 3 smaller pieces. This restriction enzyme cut it into 3 smaller pieces. Pretty cool. And the numbers of times the restriction enzyme will cut for one human is different for another human. Why is this important? Well, we can take DNA that's been treated with a restriction enzyme. We can run it out on a agarose gel and separate the DNA pieces by size. And what will happen is the smaller pieces will go faster than the larger pieces. So larger pieces will move a certain distance, smaller pieces will go even faster. This lane here is just showing some different sizes. So bigger, bigger, smaller, smaller, teeny tiny, way down there. Now this can be used in DNA fingerprinting. So in DNA fingerprinting, you can analyze DNA that's found, say, at a crime scene and try to see who that DNA belonged to. So we have some size lanes showing different sizes of DNA. This is the control. We got control DNA showing the restriction enzyme works. Then we have DNA from the victim, DNA from suspect 1, and DNA from suspect 2. We treat them with the restriction enzymes. Victim leads to this size of DNA. Suspect 1 gives us these 2 pieces. Suspect 2 gives us this size piece. Then we have 2 DNA samples from the crime scene. DNA sample 1, treat it with the restriction enzyme, run it out, and oh, look at that. It matches the victim. Who would have guessed the victim would be at the crime scene? Then you have DNA sample 2, treat it with restriction enzyme, run it out, and oh, look at that. We get this pattern. This pattern matches, look at that, suspect 1. So suspect 1 was at the crime scene. May even have been the person to commit the crime, but maybe not. So again, as you can see, it's possible to take DNA unknowns and match them to people to see who in this case was at the crime scene. This system can also be used in paternity cases. So, as we know, half of the DNA comes from mom, half of the DNA comes from dad. And here is where they ran out the DNA for the child, got a certain set of bands after it was treated with restriction enzyme. Took mom's DNA, treated it with restriction enzyme, got a different set of bands. However, you'll notice that some of the bands overlap between the mom and the child. This makes sense. Almost always know who the mom is. Then we have dad. Dad's DNA is cut with restriction enzyme. So, looking at the dad's band compared to the child's, is this dad the biological dad? No, not the biological dad. He could still be a great loving father, but he's not the DNA dad. Here's another case. We have mother, son, daughter, and father. Their DNA has been treated with restriction enzymes, run out of jail. So what do the results show? I'll pause a moment, try to figure that out. Feel free to pause the video if you need more time. All right, it turns out that the son is indeed biologically related to the dad. The daughter is not. So again, here's the father. Here's some of his bands. Here's a band that matches with the son. Here's another band that matches with the son. Another band that matches another band. Looking at the daughter, not really that many matches. But again, he could still be a wonderful, loving parent to both of the children. All right, and that is it for this lecture of Lab 3.